Okay, so we're talking about the cell body of the neuron. It is the biosynthetic center and it's got all the normal organelles except that it doesn't have centrioles. Remember that centrioles are needed for cell division and neurons can't divide. They're amitotic, so they're absent in the soma or the cell body of a neuron. Just like we had endoplasmic reticulum or rough ER, or we called it sarcoplasmic reticulum in the muscle cell, we have rough ER that has what are called nesal bodies or chromophilic bodies and ribosomes in the neuron. These areas are very, very active, constantly making new proteins and then sending them from the cell body down into the extensions of the cell body, the dendrites and the axons. The Golgi apparatus is very active and it actually forms a circle around the nucleus. There are increased mitochondria because you're going to need lots of energy to fuel this very active cell. As we make this part and make energy, we have to have lots of microtubules that are like passageways down the axons and dendrites. Some of them are antegrade, meaning they only go in one direction. Some of them are retrograde that come back. So to communicate with these long extensions and send cell substances down, we have a very elaborate system of microtubules. Now, cell bodies are clustered together in the central and peripheral nervous system, and so where you have a cluster of cell bodies in the central nervous system, that we call that a nuclei or a nucleus. Where we have clusters of cell bodies in the peripheral nervous system, we call that ganglia. Here's a picture that shows you the lentiform nucleus, so you know that this is a group of cell bodies of several neurons. Notice it's a little darker in color than this out here. This is actually gray matter. Ganglia, if you notice that you have a neuron and it says the preganglion neuron, and here is outside of the central nervous system, so all these cell bodies would be together branching off of different neurons branching off of this area. So this area where cell bodies are found in the peripheral nervous system are called ganglia. Most cell bodies are in the central nervous system so that they can be protected by the skull and the vertebral column. So let's talk about the extensions or the processes out of the cell body. Extensions, just like cell bodies, run together. Have you ever seen somebody cut a telephone line and you see all those hundreds of individual telephone lines and they're all packaged together? Well, when these extensions run through the body, they're going to be packaged together. In the peripheral nervous system, this package of extensions is called a nerve. So when I say my sciatic nerve, it's really lots of axons together bundled. In the central nervous system, this bundle of extensions is called a tract. So we have nerves in the peripheral nervous system, tracts in the central nervous system. So let's look at the two types of processes we have. We have dendrites and we have axons. Usually there's more than one dendrite attached to the cell body. We're going to see that there are different kinds of neurons, and there's one neuron that just has one structure going in. But this one that's shown here in the bottom is a typical neuron, and so you can see there's more than one. Short branching, and they do that to increase the surface area, so the chances of picking up on an incoming impulse and sending it down the line are greater. It is part of the receptive region or the input region of the neuron, and it directs its impulses towards the cell body. The kind of potentials that come in dendrites are graded potentials. That means that they're going to only travel a short distance. They can add up to be an action potential, but they're very short and they can die out. Let's say in order to fire the action potential down the axon, I need 75 millivolts. Maybe 2 millivolts came here, 20 millivolts here, and 50 millivolts there, and 4 millivolts there. They could all add up to send an action potential down the axon. It's kind of like adding up all of your grades, and if it's enough, you pass. So these are graded potentials. So these are the dendrites. This is the cell body, and this is the receptive region. The potentials here are graded. They're short-lived, but they can add up to be an action potential. So let's look at axons. Axons begin at the axon helix, or what we call the trigger zone. They can be short, they can be absent, but most of the time they're very long, and we know them as the nerve fiber. They can branch at right angles. You see this one branching at right angles. Those are called collaterals. And the end of the axon, there are axon terminals, and actually down here we can branch very extensively, 10,000 or more of these little terminal branches. Sometimes axon terminals is the way we refer to them, sometimes synaptic end bulbs, and sometimes boutons. The axon is actually the conducting component, and later the axon terminal is going to be the secretory component. When you have enough voltage to fire the impulse down the axon, this is the conducting zone, the conducting that conducts the impulse down the axon. And then this last region, because the impulse is here, neurotransmitters will be released or secreted. So this is the secretory region. So the note says conducting component to generate and transmit action potentials from the trigger zone to the terminal end. And here you can see three different types of neurons, but you can still see you have the receptive region, the conductive region, and the secretory region. Here again, you have the receptive region, the conducting region, and the secretory region. So the secretory region is at the very end of the axon. This is where you're putting the signal to the next excitatory membrane, so it's the output. The impulse reaches the terminal end and causes neurotransmitters to be released. Now this is a little bit different than we saw in the muscle cell. At a neuromuscular junction, there was only one kind of neurotransmitter released, and that was acetylcholine, and it was excitatory. But in the nervous system, when you're having a nerve innervate with another nerve, the pre-nerve, presynaptic nerve can actually secrete a neurotransmitter that inhibits the signal or slows the signal down. So we have many types of neurotransmitters in the nervous system. So again, we have the receptive region, and this particular neuron, you see the dendrites in the cell body. Right here is the trigger zone, or the helix. If the voltage is high enough to cause an action potential, it will be conducted down the axon and then go into the secretory region where we release neurotransmitters. Now, in the axon, there are no nasal bodies or Golgi. So in order to get products that the axon needs, we have to send it down those microfibrils we were talking about earlier. So there are going to be some that are moving down the axon. Those are called antegrade. And then there are some that move back up towards the cell body. Those are retrograde. And so we have communication through microtubules, since the axon has no way to regenerate and package up. We're relying on all of those substances from the cell body. Now, some axons can have a myelin sheath associated with them. Look at this axon. Do you see those little white blobs? That's fatty substance called myelin, and that myelin is going to insulate that axon so the impulse travels faster down it. So myelin sheath, there is a white fatty sheath that is segmented called a myelin sheath. It covers long, large nerve fibers, or axons. Myelin protects and insulates to increase the impulse speed by 100 times. Remember, in the peripheral nervous system, we're going to be making it in segments by the Schwann cell. So the Schwann cell is going to, or we called it a neurolimocyte, is going to make individual little segments of myelin, and then the spaces that are between the segments, those are called the nodes of Ranvier, or neurofibril nodes. They have no myelin over them. They just have the neurolemma. So over the myelin is the neurolemma. So the neurolemma is here, and then it would come down over the segment, the node that doesn't have the myelin sheath. Remember, the sarcolemma was the membrane around the muscle cell. Neurolemma is around the nerve cell, or the extensions of the nerve. In the central nervous system, remember it was oligodendrocytes that made the myelin, but instead of making little segments with nodes in between them, they're making great big sheaths at a time, and so there's the nodes are more widely spaced. So here you see one oligodendrocyte making all of this sheath in the central nervous system. There is no neurolemma. In the central nervous system, where you have nervous tissue that's myelinated, it makes the tissue look lighter in color, and so we call that white matter. Where we don't have any myelin, that's where cell bodies and dendrites mostly are, but we do have some unmyelinated axons too. That's called gray matter. It really looks more beige and brown, but the beige, the lighter color is white matter, so this is going to be very fast impulses. And then cell body and dendrites, where they're receiving messages or slower conducting axons, are in the gray matter. And so you can see tracks of white matter in the brain and going up the spinal cord. See the descending and ascending tracks, those would be white matter. Here you can see white matter. So how do we classify neurons? We can classify neurons by what they do and what they look like, the structure. We have 99% of our neurons being multipolar. That means that if you go to the cell body and you count the extensions, it's more than 2. 99% of your neurons are multipolar. There are some very rare bipolar neurons found in the retina and the olfactory mucosa. Going to the cell body, we only have 2 extensions. And then we have some that are very rare called unipolar, where actually the dendrite comes in the same extension that the axon goes out of. So there's just one pole. So this is a table that's in your book that reviews those 3 types: multipolar, bipolar, and unipolar. And notice it says that there's one extension. The cell body forms a central and peripheral process. They comprise an axon. So we can also name neurons by their function, how they work. Sensory neurons are going to be afferent. They're going to receive the information from the environment and send the information possibly to the brain and the spinal cord. Motor, or efferent with an E, are going to be transporting messages away from the central nervous system to an effector, either a muscle or a gland. So exiting is efferent, going towards is afferent. Now, neurons that are found in the central nervous system are called interneurons or association neurons, and it connects the central nervous system together. 99% of the neurons are going to be interneurons, or 99% of the nerves in the central nervous system are interneurons. And we're going to stop there before we go into the physiology.